Dear students, please note down. The notes of this video lecture are available in the description of video. Please download your notes. Dear students, today we will discuss ultrastructure of nucleus. Note down, ultrastructure of nucleus. This is an important topic for all competitive exams. BSc students, first year students, this is final exam topic. In today's lecture, we will cover ultrastructure of nucleus that shows following 5 parts. This outer double membranous coat, it is nuclear envelope. Just below this nuclear envelope, a fine thread-like network is there. This one is fibrous lamina. Very important question for competitive exams, fibrous lamina, or it is nuclear matrix. This thread-like network which is present in this nucleus, it is chromatin. Third part is this small rounded without any membrane, it is nucleolus. And the sap, the fluid which is present inside the nucleus, that is nucleoplasm. It means in case of nucleus, we will study 5 components: nuclear envelope, nuclear matrix, nucleolus, this thread-like network, it is chromatin, and dear students, this fluid, that is nuclear sap or it is nucleoplasm. First of all, we will discuss nuclear envelope. It is karyotheca. It is nuclear coat. It is nucleolemma, nuclear membranes. This is a double membranous coat. Outer nuclear membrane. And this outer nuclear membrane, it is ectokaryotheca. Dear students, it is about 60 to 90 angstrom thick. And this nuclear coat, outer nuclear membrane, this outer membrane, it is ectokaryotheca that consists of small rounded structures. These are ribosomes. Note down, competitive students, these are ATS types of ribosomes. Later on I will discuss ribosomes. These small rounded bodies which are present on this outer surface. So it is rough. It means this outer nuclear membrane, it resembles rough endoplasmic reticulums. But this inner membrane, it is smooth without any ribosomes. So this inner nuclear membrane, or it is endokaryotheca, it resembles smooth endoplasmic reticulum. Both outer and inner membranes are separated by a space. This one is, it is perinuclear space, and this perinuclear space is about 100 to 500 angstrom thick. This outer membrane having ribosomes, so it is rough, but this inner this inner nuclear membrane without any ribosomes, so it is smooth. Between these two nuclear membranes, a space is present, and that space is perinuclear space. Now down, this inner nuclear membrane, it is lined by a fibrous layer, that is fibrous lamina, or it is nuclear cortex, which is about 300 angstrom. This fibrous lamina, note down, very important question, this fibrous lamina which lies below the inner nuclear envelope, that provides a supportive framework. It provides surface for the attachment of telomeres. The ends of chromatin are embedded in this fibrous lamina. Now down, it means this fibrous lamina that acts as scaffold of chromatin, means the terminal parts, the telomeres, the ends of these chromatin are embedded in this fibrous lamina. It is nuclear cortex that is a part of nuclear matrix, which is about 300 angstrom width. Now down, this fibrous lamina, another important question, that provides material for the formation of nuclear pores, as well as this fibrous lamina or nuclear matrix that involves in the breakdown and reformation of nuclear envelope at the time of cell division. Dear students, note down, at the time of cell division means at the time of mitosis and meiosis, this nuclear envelope disappears as well as it reappears. In that process, fibrous lamina plays an important role. This fibrous lamina involves in the breakdown of this nuclear envelope, as well as during cell division it involves in the reformation of this nuclear cord. This is the vertical section of nuclear envelope. This nuclear outer coat that shows it is porous in nature. This nuclear coat, which is double membranous coat, its outer membrane that is rough due to presence of these ATS ribosomes, and this inner membrane, it is smooth without any ribosomes. This outer membrane resembles rough endoplasmic reticulum, while this inner nuclear membrane or endokaryotheca, it resembles smooth endoplasmic reticulum. This outer membrane, it remains connected with endoplasmic reticulum, and this inner membrane is lined by this fibrous nuclear cortex or nuclear lamina. This nuclear envelope shows its porous nature. It means this double nuclear envelope, it is porous. It possesses about 1000 to 10,000 nuclear pores, and through these pores, this nucleus receives information from outside, from the cytoplasm, as well as through these nuclear pores. The nuclear information is passed from this nucleus to cytoplasm. It means these nuclear pores act as passes for the molecules like proteins, like ribosomes, like RNA, from this cytoplasm to nucleus, and from this nucleus to cytoplasm. And these nuclear pores may be of simple type, may be annulated type called annulus, having a small partition, means pore with a septum, and this nuclear pore shows an electron dense plug is there, and this nuclear pore is blob type. Dear students, in next lecture I will discuss the entire structure of this chromatin. In this nucleus, this small rounded structure, it is nucleus, and this small globular structure, these are smaller as compared to these nucleolus. These small structures are called karyosomes, or these are named false nucleolus. Now down, this one is which is larger in size. This is nucleolus. But these small bodies are false nucleolus, or these are chromocenter, or may be named endosome, plasmosome, karyosome. Note down this entire network, this thread-like structure, that is chromatin, and this chromatin having two parts. This fibrous part, which is diffused, that is called true chromatin, or it is euchromatin, and these thick structures, dense structures, in which the entire chromatin is packed, diffused. It is diffused, then it is euchromatin, and when the chromatin in the form of condensed form, then it is heterochromatin. Dear students, in tomorrow's lecture, I will discuss chromatin, nucleolus, nucleoplasm in detail.